All right, so we are going down the path of glycolysis, and if we take a look real quick, we're at step number 5. Okay, that's the next step we'll look at, but I want to go back real quickly and look at a couple of the steps and describe something. We describe the mechanism, but in step number 1, glycolysis cost us energy. Okay, we used energy. And the whole point of taking glucose is to make energy, right? And so step 1 cost us energy. We use. Step 2 doesn't give us any energy, okay? But we didn't use any energy. We just converted the molecule into a different form. So this one's net nothing, all right? We didn't gain anything but didn't lose anything. And we saw the mechanism. We wrote that. Step 3. In this case, we used ATP. So we're using energy, not gaining any energy. All right? Step 4, by the enzyme aldolase, what we took was the fructose 1,6-bisphosphate and converted it into 2 molecules, one dihydroxyacetone phosphate and one glyceraldehyde 3-phosphate. Again, no use of energy. But at this point in time, where is the potential energy? The energy that we can actually draw from?
Would still be in the phosphate bonds.
There is energy in the phosphate bonds. The second place that we actually have energy is in the electrons that are attached between carbon and hydrogen. So that is a source of energy that we haven't yet extracted. And so it's still in the carbons themselves. Once we oxidize carbon all the way up to CO2, that's as much energy as we can extract. But we still have the ability to take electrons away from carbon, add oxygens to it, and potentially get energy. And so we think about the phosphate bond as a lot of energy, but there is a secondary. Now, this reaction was just a tad complex, and the mechanism is now, I showed you in class, and there's a sheet on the Moodle site that you can look at, and we have our 2 entities. Important about this particular enzyme, in all the other enzymes, we stabilize the transition state through a non-covalent interaction. Okay. We, in some way, if by chance there's a negative charge that is produced, somewhere around there inside the enzyme there's a pocket that has positive charge that kind of balances it out. This enzyme, the aldolase, actually covalently links to the enzyme to help reduce the transition state. And that's a difference between everything that we've seen up to this point in time. Step number 5, again, uses no energy but gains us no energy. But what it does is it simplifies what we have to do. Because in the last step we had 2 different molecules. Potentially the body could have had 2 different pathways to get it to where it needed to go. But what it elected to do in God's design is to take one of those molecules and convert it into the other, so that now we only have to go down 1 street, we don't go down 2 streets to reach the same place. There's an efficiency. Now, if we start looking at the names of the enzymes, it tells us what's going to happen. Kinases, which we've used 2 of them, add a phosphate from ATP. We have seen an isomerase before, and we're seeing an isomerase again. An isomerase changes the molecule, makes an isomer of a molecule, but it loses or changes no numbers of oxygens, carbons, hydrogens. The number of things stay the same. The mechanistic process is to deprotonate and protonate a number of times, usually a couple times to get to where you want to go. And so if you take a look at the top, we do some inventory again. We see the phosphate and the phosphate on both of them are the same. This oxygen is the same. This carbon is the same. But we've got differences above that. Here we have a ketone, here we have a hydroxide, Here we have a hydroxide. Here we have a ketone. All right. In this bond, we lose this hydrogen here on the oxygen and one of these hydrogens. And the product is gone. Around this bond, this oxygen is going to pick up a proton, and this carbon will have a proton at the end. So that's what we have to do. We have to move 4 protons around. Now, if I were predicting and you didn't have an intermediate, okay, you would have essentially 2 protons that you could have taken, A or B, and I'm also going to call this one B. Why can I call those 2 protons B? They're the same. They're equivalent in their stereochemistry and everything, so if I took one or the other, it really doesn't matter. Okay? Usually the enzyme takes the most acidic. Okay? The most acidic one, all right, there are 2 options here. A lot of times we think if I take a proton off the oxygen, as in this place, then the oxygen can carry a negative charge. That's relatively okay. So that proton is acidic. It can actually be abstracted. But more powerfully, okay, and you may have talked about this in organic chemistry, if you have a carbonyl, the alpha carbons become acidic because you can form an intermediate. And so in this case, if I'm going to use the alpha carbons, alpha to a carbonyl, these 2 would be acidic and those would be acidic. Okay? They're equally alpha, right? Why would B react, because obviously that's what we're going to see here, and not these 2? What would be the reason why we're going to react up here and not down there if they are both alpha to the carbonyl?
Hindrance?
It is a hindrance argument, okay? In an organic flask, you're going to have both reactions occur. Okay? If there's a base there, you're going to deprotonate some at the top one and some at the bottom. But inside the enzyme, and what we do is, we talk about there's an enzyme there, the protons here are buried inside and inaccessible, and the reaction is hindered because they're blocked by the physical protein. These protons will be in the active site right next to the entity that acts as a base. And so this is why we control how the reaction occurs. Inside a beaker, you would get both reactions and only one of them you want, and so your yield may be 50% because half of them go the other direction. There's no way to direct it. But this catalyst directs what's going on. Now, we always have inside an enzyme what to use and what to use? An acid and a base. And I'm going to use a acid and a base. And my daughter is trying to call me. All right. And so I'm going to take one of the bases, one of the protons with a base. When I do that, this double bond here now can form the double bond. Too many bonds to carbon, I can now pick up the proton, which leaves me with the intermediate state. Okay, not exactly what I want, right? I want to have a ketone in the number 1 carbon at the top. I now have a hydroxide. So what do I have to do to get from the intermediate state to the final product? Which proton do I take off? The hydroxide at the top. So I'm going to just articulate that. All right, and I'm going to have always an acid. And so my base, which is some amino acid inside the enzyme, we'll take that proton. I'll take this pair of electrons, form the double bond that is in the final product, and then I pick up a proton to become this proton that is here becomes this proton right here. And I'm now at glyceraldehyde 3-phosphate. Now, in this, there are equilibrium signs. This one's a whole lot more stable. But this one is the one that is reactive to the next step. So in this mixture, most of them stay right here. But if I get to the glyceraldehyde 3-phosphate, I'm going to react it away down to a concentration of 0. Then the equilibrium pushes over a little bit, and I react it away to a concentration of 0. And then I can drive the reaction all the way to the end product. The next step is a new enzyme called a dehydrogenase. Okay, dehydrogenase is an enzyme that can do a redox reaction. Now, a redox reaction is going to move electrons. And in most biological reactions that we look at metabolism, what we're going to see is the redox reaction is going to have a hydride that leaves. Now, if you predict a hydride leaving in organic chemistry in just about every reaction, it's going to be wrong. Okay, hydrides don't necessarily like to be a leaving group. They're a very poor leaving group. But what happens is every dehydrogenase that you see will have an electron acceptor. And because of the electron acceptor and the stabilization of the enzyme, this is why we can have a hydride leave. You can only have a hydride leave if there's an acceptor. Now, there are 2 acceptors that we initially teach, and these tend to accept hydrides. All right, and the 2 are NAD plus and FAD. Did you guys see the animation about that? The NAD usually reacts around a hydroxide to create a ketone. That's normally what happens. Okay. And an FAD normally takes a straight-chained alkyl, just kind of a carbon, hydrogen, and it'll make a double bond in that. They're just kind of optimized for that. But NAD plus can have a couple other times in which it can actually accept, and this is one of those times. And so let's watch what the NAD plus actually does in this case. Now, inventory. Okay. Here and here, the same. This is the same. This is the same. Ooh, interesting. This ketone is now attached to a phosphate. This ketone is attached to a hydride. Okay. Not just a hydrogen. You're actually going to lose the hydride itself. All right. If you look at this enzyme 2, normally to put a phosphate on, it costs ATP. This reaction is actually going to generate a high energy intermediate without the use of ATP. So I'm actually placing energy into the system that I'm going to be able to extract later. Now, this looks simple, but this is another enzyme that actually becomes covalently linked to make the transition state a little bit lower in energy. And so this is what happens. Inside the enzyme, you have the NAD, which is one of the reactants, and we have a reactive cysteine. What's important, and you should understand, you may need to go back and look at all of the amino acids and know all 20 amino acid R groups, and then kind of the category they fall into. Are they acidic? Are they negative? Are they positive at pH of 7? And are they aliphatic? Are they big? Are they small? Are they aromatic? Okay? Because what we're going to do is you justify a lot of things that happen based upon the R group of amino acids. So the cysteine has an SH as part of its R group. Inside this enzyme, the sulfur becomes deprotonated. It's a nucleophile. And so what happens is the reactant, the glyceraldehyde 3-phosphate, enters into the active site. It orients it such that the sulfur is going to react with the carbonyl. Also, that places this hydrogen and the pair of electrons close to the electron acceptor. All right. Once this covalent bond is created, remember when we attack a carbonyl, we always kick up and the electron pair is on the oxygen. This is one of the high energy states, the transition states. In an excited state, it doesn't stay that way, even though it is stabilized. I do want to reform that double bond. It's a much more stable conformation. And when I do that, the only leaving group that is effective or that makes progression in the reaction is the hydride leaving. Only leaves because there's an acceptor immediately there. All right. I have gotten the hydride off, and important, right here, that's a pool of energy in which my molecule came in, the glucose first came in, and I've converted it into a true carrier of energy. We talk about it as an electron carrier, but this is a pool of energy that the first real big conversion happens. All right, a couple things happen. The NAD+, that energy moves out into the cytosol. And NAD+ comes back in to fit into that active site, and now an inorganic phosphate will come in. Now the inorganic phosphate has negative charged ions, oxygens, and negative charges can always be nucleophiles. And so this negative charge will attack that carbonyl. Now, unfortunately, one part of this mechanism was written right, and this one is written incorrectly. I guess the artist ran out of room. Remember, when we attack, we always have to kick up, and we always have to kick down. And so if I look at this, this would be the first movement, the second movement, the third movement, and that would be the fourth movement. To regenerate the S- in the active site, and what comes out is a molecule called 1,3-bis-phosphoglycerate. Yes?
What happens to the hydrogen level phosphate?
Very good. A lot of times when we do reactions, when it is in the active site, the phosphate by itself roams around because of the pH in the system with a proton on it. It is an argument of pKa versus the pH in the system. Okay, and you can talk about if the pKa is higher than the pH, you get protonated, those kinds of deals. When the phosphate is attached to the carbon, it changes its pKa. And so when it's released, there's a lot of times that protons are just deprotonated because of the solution surrounding. It's not an enzymatic reaction, and so quite often something will fall off and then it changes pH state, and it's assuming it's going into the cytosol at a pH of a little over 7. And so you'll see a lot of that happen, where you'll show it, or you'll articulate that there's a proton on it, and then when it leaves, it's deprotonated all of a sudden. And so what it's going to do is, hopefully the artist will always show the protonation state that it's in at a pH of just a little bit about 7, although sometimes artists are not chemists, and they just they will write it incorrectly. But the 1,3-bisphosphoglycerate is what the protonation state would be expected. All right. The other thing about this particular reaction is it happens twice, because remember, we took one glucose and made eventually 2 glyceraldehyde 3-phosphates. So this goes twice. So I make twice as much energy in this step because I have 2 NAD pluses created. 2 NADHs created. Step number 7, okay? This is a kinase, okay? But this kinase is a little bit different than you expect. A kinase actually transfers a phosphate. We normally add a phosphate to something from ATP. But what we're going to do in this case is take phosphoglycerate, the 1,3-bisphosphoglycerate that we just made, and react it with ADP to create ATP. The transfer of the phosphate is backwards than what we would have expected. And I'll be left with a 1,3 phosphoglycerate. Now, to do this, what in the world do you think is the nucleophile? What attacks what to do this reaction? If I look at this, inventory, all of this is the same right here. On this oxygen, there's a phosphate, and it's lost. Oh, wait, where did it go? It went right up here. So that phosphate has to be bound to that oxygen. Okay, the nucleophiles are the negative ones. So to get that phosphate, which is represented here, attached to that oxygen, which is represented there, this oxygen needs to attack it. No base is required at this one because it's already deprotonated. And so this negative charge attacks the phosphate. Okay. When I attack a phosphonyl, what do I always have to do? Kick up, kick down. And to break the bond between this phosphate and that oxygen, I articulate it this way. I take this pair of electrons and I put it right onto that oxygen, and that breaks the bond. So it is a kinase. It transfers the phosphate, but it did it in a backwards mechanism than what we would expect. But you can see where it starts and where it ends, and you can see why it transfers that way. We have just created energy. And again, because this is done twice, we create 2 ATPs at this moment in time. In terms of the stoichiometry of the ATPs, at this moment in time, steps 1 and 3 used 2 ATP. I just created 2 ATP. I've made 0 ATP. But I have made NADHs that are energy molecules. All right. Step 7 was wonderful. Was able to get some energy. Okay. And, here, to tell you, you have 2 ATPs. That's a waste of a slide, but it's there. All right, step 8. The 3-phosphoglycerate, can't use it. So I have to change it into something called 2-phosphoglycerate. All right, so why in the world is it called 1 and 2? A 3 and 2. This is the 1 carbon, this is the 2 carbon, and that's the 3 carbon, so 3-phosphoglycerate. Moving to 2. All right, that seems so simple. I just move it. Just shifts right over, right? Oh, it's never that simple. Okay. What is interesting is that this phosphate here in blue makes, and that phosphate in green are 2 different phosphates. They are not the same phosphate. You would never have known that. All right. Also, this particular enzyme is a mutase. It's going to make, change the molecule, and it does it in a special way. Again, the enzyme becomes involved in the reaction, and there's an intermediate. So the enzyme itself, and there's a little protein. I've given you a very detailed look at a protein. Has a side group. Okay, what is this group? What R group is this one? Okay, you got a 1 in 20 chance of getting it, right? Do you remember what this R group was? You need to look over the 20 amino acids. This is histidine. Okay, histidines are pretty special. They have a pKa of about 7. You're going to see them used quite often in biological systems. They're very active at pHs that are in the active sites. A lot of times they will act as either an acid or base because they can be protonated or deprotonated, but they can also be reacted. And in this case, the enzyme, for it to be functional, has to, at one moment in time in its history, add a phosphate to the histidine. And so the 3-phosphoglycerate comes in, and it interacts with the active site that has a histidine that is phosphorylated. What will happen here is I'm going to create an intermediate that is the 2,3 bisphosphoglycerate. This one is never seen inside the cell. It is only converted and made inside the active site. It cannot be released. What can be released is the 2-phosphoglycerate. And so we're going to add a phosphate. If you take a look at inventory, that oxygen starts off with a hydrogen but now has a phosphate. The rest of it is the same. And so now I've identified where the extra hydrogen is. Base takes that off. The oxygen becomes a nucleophile, and the oxygen needs to be attached to a phosphate. I will attack the phosphate. Up, down, and for a moment, the nitrogen, or the enzyme, becomes the leaving group. Now, what's important about this is that I really didn't show three-dimensional structure, okay? Because I have to have space to write my arrows, and I'm taking a three-dimensional process and putting it on a two-dimensional piece of paper. When the reaction occurs, and this is in the phosphorylated state, when the enzyme binds, this histidine is right here. Okay, it's positioned above that oxygen. When the phosphate leaves, it causes the enzyme to change shape just a little bit. Okay? And what happens is this nitrogen of the histidine doesn't sit three-dimensionally right here. It shifts over. And it sits right here. Okay? And so it's positioned in the phosphorylated state, sitting kind of right above this oxygen. And in the dephosphorylated state, it sits right above this phosphate. A lot of times, the function of the protein is just a small change in its structure, and that's not a lot of angstroms to move, and so it just moves over. And this is how it directs the reaction. And so now we can see from the product that this oxygen is a phosphate. Here it has to be hydroxide. We see that the nitrogen doesn't have a phosphate, but here it does. So what's going to happen is this pair of electrons is going to attack, kick up, kick down, and we always have a proton. And so I pick up that proton to create the hydroxide. But what has happened is the original phosphate now sits on the enzyme, and the phosphate that was in the enzyme is now in the intermediate. Now, we could have labeled the phosphate, put a radioactive phosphate on there and tried to follow it, and we actually saw that there was an exchange. It wasn't an internal attack. It didn't just move it over. It actually exchanged it. And so the next round, the phosphate that was placed now gets put on something else, and it shuttles around. Effectively, if you just look at the molecule, you wouldn't have seen that, and if you don't label it, you don't see that. But it took us a little while to figure this out, that it wasn't an internal attack. Okay, 10 steps, we're at 8, so we're almost there. All right, enolase. All right, we've got water coming in and water going out, or actually water going out. All right. Okay, you see all of that going on. But what actually really happens here is kind of more simple than you think. It's really one of the first mechanisms you learn in organic chemistry, but it looks like this. In organic chemistry, you have a textbook that'll show you this. Okay? and they say, I'm going to do an elimination base catalyzed. So they say you drip in hydroxide. The hydroxide will take the proton to create water. I will create the elimination, or create the double bond, and what leaves now is a hydroxide. And I create this intermediate. Okay? And it looks so simple there because what they've done is they've just eliminated all the R groups. They just kind of put lines. All right? Essentially, the reaction here is the same because this becomes the hydrogen that's there, and this hydroxide is the hydroxide there. I just put R groups onto it. Now, you could see that other one really easily, right? But if you identify, this is just as simple. It's just I put R groups on it. So focus in on what is actually happening. And so I've actually now given away the entire mechanism. The base will take the proton. The negative charge can be placed in the double bond, and this can fall off as a proton. All right? Now, what is interesting about this enzyme is that 1, 2, 3, not exactly how it does it, because when you do this reaction, the first one, that first move, it creates a negative on a carbon. That's really high in energy. This enzyme particularly has metals that sit up here, and I believe they're magnesiums, so 2 plus magnesiums. Okay? And so there's an intermediate step where one actually does happen, but before it goes down, what actually happens is I have that as an intermediate where I get the negative charge on that oxygen. Negative charges on oxygen are better, and they're even better because these negative charges here interact with the magnesiums. And so they're locked in with a lot of positive charge, and I create an intermediate that has less energy. Now, to finish that reaction, again, it's in the higher state, can't be in that excited state all the time. I fall in to create this. There was a double bond that was here that creates that double bond, and then I leave. But if I didn't tell you that there was magnesiums and that it was done that way, I would have accepted the 1, 2, 3 green ones originally. And we get to something called phosphoenol pyruvate. All right, phosphoenol pyruvate has a pretty great acronym, PEP. Okay, and phosphoenol pyruvate is going to be seen a lot more often, okay, from this point on. The last step, pyruvate kinase. All right. Have you seen something similar to this? Yeah, we just saw one, right? We just saw another kinase where the ADP was the attacking group to make ATP. And so there is a lot of repeats in what we're looking at. And so we know I've got to get that phosphate on there, and I have to attack what would be the beta phosphate onto that phosphate in the phosphoenol pyruvate. But what's interesting is what comes out. Okay, what is the end product? It's actually a tautomer of what we would expect. All right, and what happens here is the phosphate will attack. I always have to kick up. I always have to kick down. Okay, that's That one you should get 2. But now it gets kind of crazy. What in the world? All right, that oxygen, usually what we do is we put the negative charge on that oxygen, right? But I don't have a negative charge here. I have a double bond. So instead of that negative charge sitting on the oxygen, I've got to put it to make a double bond. But that's too many bonds. Well, what we'll do is, that means I have to push something out, and that double bond is not there anymore. I have 2 hydrogens. I have 3 hydrogens. And so what's going to happen is that last movement off of here will actually put that double bond there, and then this will pick up that proton. And I have another energy molecule, 2 of them, because I've done this twice. And the balance of energy sits in pyruvate. And remember, the numbers, I've only extracted 85 of over 2,800 kilojoules through all of these processes. So the majority of the energy still lies right here in pyruvate. All right? And so the 3 pools of energy that we have at the end of glycolysis, all right, are the pyruvate, which is big, okay, the ATP, which is what we think about, and the NADH. That's where our 3 pools. Now, we're going to, through all of this reaction, eventually get them to ATP. We're not going to make a lot more ATPs. Yes? No, because we had to use 2 ATPs originally. And so, yes, we did make 4 in the process, but we had to take account the first 2 steps. All right? That's not going to grow too much until we get to the last step. Okay? And before we can actually get it to ATP, we're going to convert it into another pool of energy. Okay? What is going to grow is this, electron carriers. That is going to be the larger pool as we go through metabolism. And then this one is going to get smaller as we convert all 6 of those carbons into CO2. Okay, so this grows little until the last step. This grows a lot, and this one gets smaller as we go through. So the carbon intermediates actually have less and less and less and less energy as we go through the process. Okay. Now, these are the steps. What is important about a process, this is a road, right? There are 10 separate enzymes. Every enzyme that you've learned in the past can be regulated, can be shifted to be faster or shifted to be slower, can be turned on or off. All right. In a process that has 10 steps, the complexity of controlling every single enzyme is prohibitive. Okay? So a lot of enzymes are controlled by this. How much product do they have? Or how much reactant do they have? If they have more product, they'll go in one direction. If they have more reactant, they'll go in another direction. So many of our enzymes just are sensitive to how much concentration are there. And many of our pathways are controlled and can be controlled overall by as little as 1 enzyme. Normally we call that the committed step. In glycolysis, of the 10, there are 3 enzymes that are control points. The reason is that glycolysis, the exit from glycolysis, is not always pyruvate. There are other places to exit, and so we have a little bit more refinement in how we control the process. And they usually occur at rate-limiting steps or irreversible reactions. And so the hexokinase is controlled very, very tightly. in the process of the degradation, and that's one side. The other is synthesis, which we will look at later, called gluconeogenesis, and I have another enzyme that gets regulated. The conversion to fructose bisphosphate, the phosphofructokinase 1 is a control point. Okay. And then the last step, the pyruvate kinase, is a control point. In the synthesis portion, actually you have to have two enzymes to bypass that because this is such a huge energy molecule. And this one is important in starvation because sometimes we use this to make glucose back from other things. We can make phosphoenolpyruvate from other things and use this to make sure our brain has enough fuel. Okay, and so these are the ones that we look at in detail. The first and kind of the going down the pathway is phosphorylation of the glucose. Once glucose is phosphorylated, it's kind of trapped inside the cell. And so we eat something, it gets into our bloodstream, it can be processed into cells using transporters, and they're called glucose transporters, obviously, and you can fill the cells with lots and lots of glucose. The hexokinase in muscle cells, and a lot of times we reference metabolism in muscle cell, and that's what we kind of reference to, and then we compare it to liver, which is a totally different process that we use. But in muscle cell, once it gets glucose, it doesn't want to let it go, and so it phosphorylates it. Once glucose is phosphorylated, it can't go through the transporter. It's trapped inside the cell. It commits it to being inside the cell for either one of two purposes, for breakdown immediately for energy, or two, for storage. Okay. In the hexokinase, there are actually 4 isozymes. There are one predominantly, the isozyme 1 is called hexokinase, and isozyme 4 is predominantly found in liver, and it's called glucokinase because they act differently. And so we look at the activity of the enzyme as a function of the concentration of glucose. And you can see glucokinase and hexokinase are different. Okay, looking at that graph, what does that graph tell you? Okay, there's a lot of graphs like this in biochemistry. You look at it, you see 2 curves. What does it mean to you? Compare, contrast hexokinase and glucokinase.
The hexokinase suddenly spikes and then levels out.
Mm-hmm.
And then the glucokinase is kind of a steady increase.
Mm-hmm. If I take a look at this, all right, normal blood concentrations, okay? The hexokinase at even normal blood concentrations is working as fast as it can. So at normal blood concentrations, if glucose gets inside the cell that has hexokinase, like the muscle, it's always phosphorylating, okay? But what about glucokinase at normal blood levels? If glucose gets inside the cell of liver, will it be phosphorylated? Well, maybe. The enzyme's only working at 25%. So for one in every 4 glucose gets phosphorylated. It seems like liver doesn't want to trap glucose. Liver wants glucose to pass through. Okay. When does glucokinase actually effectively start trapping glucose? What has to happen in the body? It has to be way too much glucose. You have to eat a Snickers bar, okay, for the concentrations to get up in your blood really high for the glucose to be phosphorylated so it can be sent down glycolysis or a pathway inside the liver. But in muscle, you eat or you don't eat, it's always trapping as much glucose as it gets in there. All right? And so what we do is we compare things at the same concentration, and we can tell the difference. Hexokinase works faster, glucokinase works slower. And that's what we should be seeing in this. Actually, this kind of, in terms of the Michaelis-Menten type of graph, this enzyme right here is a true Michaelis-Menten. This is kind of an allosteric, and we're going to see a sigmoidal shape to it. It's a little bit more complex. All right? And so there are two different pathways. And so the amount of starting material for any pathway is kind of a rate-limiting step. If you can't trap a lot of it, your whole pathway can't work very fast. All right? And that's one thing about hexokinase. All right, the Km is 0.1 millimolar for the hexokinases. What is a Km? I'm saying this like you know what it means. Do you remember? Ah, yes, it's 1/2 Vmax. In terms of the enzymatic equations, okay, Km is 1/2 Vmax. All right, Vmax is the maximum rate that that enzyme can work. All right, and really what it's saying is half the active sites are filled. So it's a good estimate of how much concentration it takes to fill that active site. For glucokinase, the Km is 10 millimolar. So that is 100 times larger than hexokinase. So it takes a whole lot more glucose to reach half activity, all right, than the hexokinase. And so we saw that in the graph, and here is the numerical values. Hexokinase itself is inhibited when glucose-6-phosphate concentrations actually go up. And so if inside your cell glucose-6-phosphate concentrations go up, it'll bind to the enzyme and start slowing it down. Now, here's the problem. If the hexokinase is working really, really fast, you build up glucose-6-phosphate, but you should be sending it down the pathway, right? But if it doesn't go down the pathway, that's why it comes back in and signals, hey, slow down, you're getting too much, I can't go down the pathway. Glucokinase is not inhibited by glucose-6-phosphate. We really never get glucose-6-phosphate up to the level, but that's one difference in the shape and the function between those two enzymes. All right. All right. In this, also glucose affects the pancreatic cells. And so glucose goes through a glucose transporter, in this case glucose transporter 2. And so anytime that has a number, there's 1, 2, 3, 6. Okay, this is just the one that's in the pancreatic cells. And glucose gets in there, and this is one of those other isozymes. Glucokinase is phosphorylated. I get glucose-6-phosphate, glycolysis starts happening, and I start making ATP. That's great. Now ATP goes to a channel, a calcium channel, and when the calcium channel binds ATP, it inhibits it or closes it. Right? Now, this depolarizes the cell, just like a neural signal. That depolarization then affects a calcium channel. The calcium channel opens. Calcium is very, very high. Here, calcium is very, very low, generally in the cytosol. So calcium floods in, which then causes a vesicle to merge with the cell membrane. And what's inside here? It's a little tiny protein called insulin. This is how the pancreatic cells detect glucose and send insulin out, seeing that we are now have a lot of energy molecules, which then makes all the cells start to signal a process really to use that glucose. And so because of the process of the glycolysis inside the pancreatic cell, that production of ATP is not just for energy, it is also a regulatory signal to tell every other cell, hey, we're seeing glucose. Start working. It'll say, start synthesizing, start making ATP, and start storing as much as you can, because it's not going to last. When you go for blood work to the doctor, and they're going to take your blood work and they're going to see if you're diabetic or not, what must you do, or what must not you do, that doesn't sound right, what can't you do before you get your blood work done? Right? You have to have a fasting state. All right? This is the reason. As soon as you eat, your blood glucose concentration skyrockets really high. Okay? It actually gets high enough that if a diabetic would have that concentration, people would be worried you're going to go into a coma. But what happens is, under normal instances where your insulin is working and all the receptors are working, your concentration goes really, really high, but then it drops right back down to the normal because all your cells suck up that glucose in response to the insulin. And that's the normal process. In a diabetic's case, is when they eat and it gets really high, it doesn't absorb the sugar, and really, really high states for an extended amount of times is dangerous. And so, but us, we do spike and come down. What they want to show is if you've been under starvation state for a long time, does your body balance and keep the glucose concentration at the right level without the absence of glucose coming in? Are you using your stores right? Are you using the synthetic pathways to make glucose? Are you signaling the use of glucose for your brain and other entities for some of the other cells that are important? And that's what they're trying to check to make sure that you're doing that well. And are you clearing glucose, making sure that it gets inside the cell? Do you have a lot of glucose in your blood? All right, so inside the liver, we have, again, glucose comes in through the glucose transporter 2, and glucose gets inside the cell. Now, the hexokinase 4, remember, has the name glucokinase. And when glucose gets high inside the cell, all right, what it does first is it comes over here and it upregulates another control point. The glucokinase, or the version 4, without glucose actually isn't in the cytosol. In the muscle, the hexokinase is always in the cytosol. Without glucose, the hexokinase binds to a regulatory protein, and it actually is in the nucleus, where it is separated from glucose. All right? The sugar actually makes this bonding separate. The hexokinase can now move inside the cell, and the hexokinase can convert this to glucose. Now, here's the thing about the liver. The liver very rarely ever goes through glycolysis. What actually happens is this glucose almost exclusively gets stored in the liver. That's where our main use of glucose inside liver. This step right here is the first step in glycolysis. If I am now have so much glucose in the liver and my storage is complete, I might actually start glycolysis. And that's a bad thing, because this particular glycotic intermediate says, hey, wait a minute, I'm not supposed to be here. It will signal hexokinase to bind back to this regulatory protein and to be put away, because even in the presence of glucose, because they don't want to waste the energy. The liver doesn't want to waste the energy and that potential glucose, making it go through glycolysis. So there's an extra layer of regulation for liver and the glucokinase, or the version 4. So hexokinase is the starting material, and if you don't have the starting material, you can't go down the pathway, so that's an important one. The real big regulator of the throughput in glycolysis is the phosphofructokinase itself, and that's version 1. We'll get to why we have a version 2 in just a second. And so remember, we're going to convert fructose 6-phosphate into fructose 1,6-bisphosphate. So we're adding a phosphate onto the position number 1, the carbon number 1. It is regulated by a couple things, both positively and negatively. if I take a look at the negatives, all right, one should be obvious. One of the end results of glycolysis is to create ATP. If I have high concentrations of ATP in the cell, don't waste the glucose breaking it down. Why don't we store it for when you don't have ATP? So ATP high concentrations will slow it down. The next one, which you don't know about, or I haven't introduced, but you may have seen it somewhere else. The next step in the process is going to take the carbons of the sugar that has been converted into pyruvate and make them into citrate. And so the next step, or the next cycle, is going to make the citrate, or the citric acid cycle. If by chance you're sending all glycolysis into citrate, right, which should go through a cycle, and the citrate starts building and building and building and building, that means the next pathway is being blocked up. It's kind of a traffic jam. And so if that particular concentration goes up, that means, hey, stop feeding the next cycle, slow down glycolysis. So that seems pretty logical about that. On the other hand, to speed this up, if by chance you have inside the cell AMP or ADP, these are indications that you don't have a lot of energy. Okay? You want to have lots of ATP around, very little ADP and AMP roaming around. But if ADP and AMP concentrations go up, that's a sign that you need to break down molecules, food molecules, into ATP. Glycolysis will be sped up. The most powerful regulator, though, is this molecule right here, fructose 2,6 bisphosphate. It's really similar to fructose 1,6 bisphosphate. The 2,6 version is not a glycotic intermediate, but it is only a regulator. And so it will powerfully make the glycolysis speed up very, very fast. And so creation of that particular molecule is important. How the ATP or ADP works is this. The hexokinases dimerize, so two of them are together. There is binding sites for ATP because that's a reactant. And so this binding site and that binding site are the normal reactant places. And you can see them in both versions. But there is a second site. This has a low affinity. This has a high affinity, meaning that really tiny concentrations will be able to bind there. But there's a binding spot here that has a high affinity. I'm going to go and make those different colors. Okay? They will only bind if ATP gets really high in the cell. And what it does is it separates the two enzymes, putting it into the downregulated state or a slower state. The enzymes, as they come together, can bind ADP, which enhance their activity. And so ADP binding there holds them together. ATP pulls them apart. If they're together, they're faster. If they're pulled apart, they're slower. And so it's a binding issue. All right. There's also one other thing that inhibits glycolysis, and that is high levels of protons. If by chance you're exercising and there are high levels of protons being formed, what that means is that you're not sending glycolysis down the proper way in an aerobic way, extracting lots of energy. You're going through some fermentation. We'll take a look at that in just a moment. And you're making lactic acid. It's an indicator to the cell, even if you need energy to slow down because you're not doing it efficiently, and so that indicator will shut down all the metabolism processes, including glycolysis. We talked about citrate, we talked about ATP. Fructose 2,6 version, the 2,6 version, okay, makes the phosphofructokinase 1 active, or the PFK1 active, and it is huge. All right. We said that ATP makes it slower, and the curve indicates that it is slower, meaning to reach 50%, it takes a lot more concentration. Okay. ADP makes it faster, meaning at a lower concentration. And so what we would do to compare the two is we take one line and it's not working very fast here and it's working very fast there. This one's an activator, this one's an inhibitor. But look at what the 2.6 version does. It makes it book, okay? And it really upregulates it. So a tiny amount of this floating around makes that enzyme work really, really fast. It is actually a feed-forward modulator, where you've heard of feedback inhibition. This is a feed-forward excitation, or making it go faster. It was discovered relatively recently in 1980, and so it was something that we didn't know for a long time, but now we find it's very, very important. Now what's interesting about this is where the fructose 2,6 bisphosphate comes from. And now what we said was the fructose 2,6 bisphosphate will upregulate, make go fast, this enzyme, and that makes more of this, the 1,6, the next step. This is also a very powerful regulator that downregulates the opposite reaction. And so it has dual control. It makes sure everything goes down and breaks down glucose, and it makes sure nothing gets synthesized in terms of glucose. Everything goes down. Where this is formed comes from the glycotic intermediate fructose 6. Okay? It will react with ATP to make the 2,6 version, and that is done by an enzyme called phosphofructokinase version 2. Okay? And so, fructose 6-phosphate has two pathways. Oh, sorry. Goes to fructose 1,6 bisphosphate. This is glycolysis. Or it could go fructose 2,6 bisphosphate, and this is the regulatory. Okay? What does this is phosphofructokinase 1, and this is the, oh, sorry. I said it right and wrote it incorrect. Phosphofructokinase 1 and the phosphofructokinase 2. This molecule, okay, goes and does this. Comes over here and makes this go big time. All right, so I actually use the same reactant, but I create two different products. Okay. To get rid of the phosphofructokinase, that's 2,6, I use fructose bisphosphatase, and a phosphatase takes off a phosphate. So I can make it with the kinase, and I can take it out with a phosphatase. Both of them are version 2. Now, interestingly enough, when we talk about enzymes, we normally talk about an enzyme has one active site, and it does one reaction, and it does it all. The phosphofructokinase 2 and the the fructobisphosphate 2 is on the actually same protein. Okay? It is a protein that has two catalytic sites. One puts on the phosphate, one takes off the phosphate. All right. That doesn't seem really good. In biological systems, we never want to put something on and take it off, or we get in a loop where we're just using energy. Okay? And so, there's a regulatory domain on this one protein. How I manipulate this regulatory domain will turn one of these on and one of these off at the same time. And so, I do something here to make one active and one inactive, or I do something here to make this one inactive and that one active. And so dual control happens with the same signal. What is that signal? In regulation of metabolism, we always have we always compare and contrast two things. And then we have a third that's an that's kind of an exception to the rule. Here are the two things. Glucagon. It's the signaling hormone that says, I'm in starvation state. I need food. And insulin. The signal that says, I am well fed, I have a lot of potential energy, start working. And so what happens? If I have insulin, I'm in a well-fed state, there's a lot of glucose roaming around. Okay? What I do is, to this enzyme, I take off the phosphate and I make the dephosphorylated form. On the regulatory portion, 1-hydroxide, I take off the phosphate. That makes the kinase active and the phosphatase inactive. This will make plus the fructose 2,6-bisphosphate. It'll make that. And then that controls glycolysis, it makes it go fast. On the other hand, if I have glucagon roaming around, meaning I'm in a starvation state and I don't have a lot of glucose roaming around, I want to save it. So what this does, it has an enzyme that'll put a phosphate on the regulatory molecule. It makes the phosphatase start moving. That means I lose the fructose 2,6-bisphosphate and glycolysis stops. And actually, later, it actually signals to make glucose. So when I have glucagon, I turn everything on to make glucose because I need it, and I turn everything off that breaks glucose down. The last enzyme is pyruvate kinase. Pyruvate kinase has two versions, a liver and a muscle. Okay? The reactions, or the control points, is an allosteric regulation in both muscle and liver, and then a covalent regulation inside liver only. So let's take a look at the allosteric. And remember what allosteric means. Something binds to the enzyme and makes it go faster or slower. That's not in the active site. And so here is the enzyme. It goes from phosphoenol pyruvate to pyruvate. That's what the enzyme does. And look, there's a lot of things that either make it go fast or make it go slow. Okay? What makes it go fast? The 1,6-bisphosphate. Lots of starting material will come up and make this one go faster. Okay? So feed forward. Pyruvate can be changed into alanine, and or alanine is being sent to the liver or sent to the muscle because protein is being broken down. Okay? If the cell senses I've got two sources of energy, glucose or muscle, and muscle doesn't start to be released until you've exercised for a while, it'll do this. It'll say, let's use the muscle that's been mobilized, the protein, and save the glucose. And so if there's evidence that protein is being broken down, it'll say, stop breaking down the glucose because this can still go back up to glucose if I need it to be. So if I have sources of protein, it'll stop that last step. Okay? ATP. Glycolysis makes ATP. If there's lots of ATP, stop it. And here's another thing. Fats. If the cell senses I have glucose or I have fat mobilized, okay? So if inside the cell you get fat in there, that meant it was in the adipose tissue, it got broken down, got put into the bloodstream, and went all the way to the cell. If you're actually seeing fat inside the cell says, don't use glucose, use fat. And so if I get fat in there, it stops the use of glucose. In actuality, if you actually do a lot of exercise, a certain amount of exercise, you usually use carbohydrates first. And they're pretty quick. You get a lot of energy from them very, very fast. But you can get really tired. And so runners feel this. They're like, they start running, and then they get a little tired. And then all of a sudden, hey, this isn't so bad. And they start running again. sometimes people call that a second wind. What really is happening is this. The body changes from a little tiny store of glucose, carbohydrates, and you start running that down to shifting over to using proteins and fat. And when you shift your biochemistry to use proteins and fat, then you can start extracting energy better, and you feel like you've got more energy because you're changing over sources. This is one of the reasons why they talk about when you exercise and you're trying to lose weight, they tell you to exercise, but they also tell you to exercise a certain length of time. Because if you only exercise a very short period of time, you'll only use the glucose stores, and what'll happen is you'll use what you eat, but you won't store any more fat. So that's great. But if you really want to burn fat, you've got to go until your biochemistry shifts, your metabolism shifts from using carbohydrates to actually using fat, which takes a certain length of time, and your conditioning actually changes that time where it shifts over. And so you have to exercise long enough to actually start utilizing those fats and breaking them down. And so the signal of protein and the signal of fat will stop the use of glucose. This happens both in liver and muscle. In liver only, we've got some other indicators, and again, a signaling from a hormone. The hormone actually causes the pyruvate to be phosphorylated. That's a covalent modification. It uses the enzyme called PKA, protein kinase A, and that will turn it off. All right. In actuality, this protein phosphatase will be signaled in insulin. I don't know why they didn't put insulin in there, but if insulin is being released, will allow the pyruvate to actually, pyruvate kinase to be placed into the dephosphorylated or active state. All right. So once we get this place where we're actually working, we go glucose to pyruvate. That's what we've shown up to this point in time. We make NADH, we make ATP, okay, and we've got pyruvate. If you are a yeast, you're actually going to take pyruvate and create ethanol. We'll look at that reaction under fermentation. If you are aerobically reacting, I'm going to make CO2 and water and a whole bunch of ATP. If you are an anaerobic, our fermentation is conversion of lactate. And the reason why we will go this way is that the reaction of glycolysis is dependent upon that. And if I can't get NAD plus back into the cytosol, you can't do glycolysis at all. So it's important to make NAD plus. But as glycolysis runs, I take all my NAD plus and put a hydride on it, and it becomes NADH. And I can run out of NAD plus. And so the process of here or there for us is a process of how efficiently I can get NAD plus back. In the yeast, pyruvate gets decarboxylated. I actually I've been to the Bacardi plant in Puerto Rico, and I've been to the Jack Daniel's distillery up in Lynchburg. The tours are actually amazingly interesting. They have these big vats that are being processed, and it looks like it's boiling. But it isn't hot at all. But it's rolling just like a boil. What happens is you're actually seeing the action of this enzyme in which CO2 is being released. Smells like, yeah, you can get drunk just smelling it. But CO2 is coming off of it. In the Bacardi plant, it was a vat that was like 3 stories tall, and you got on this ladder and you walked across it, and boy, whew, you could smell it. You make acetaldehyde, and in the acetaldehyde, what happens is NAD is usually accepting. What happens is the NADH that is produced in the part of glycolysis, the hydride attacks the carbonyl to create the hydroxide, creating the NAD plus to allow more glucose to be reacted. This transfer of the hydride and this acceptor is the endpoint, and so I can regenerate the NAD plus. Now, The problem is when this hydride attacks, the electrons have to get onto this oxygen temporarily. The enzyme is facilitated, or it reduces the energy of that by having a metal catalyst. The metal actually coordinates, and zinc is a +2 state, and so the oxygen is coordinating to the zinc, and when the oxygen receives its negative charge, it really makes a better bond to the zinc, lowering the transition state. And so the zinc metal is what catalyzes this reaction or reduces the transition state energy. In the body, if I can't feed the NADH to the mitochondria, and that's where I'm going to reproduce the NAD+, then the NADH can react with pyruvate, create lactic acid, to create more NAD+, which then gets recycled and breaks down. What this allows us to do is to produce 2 ATPs for every glucose, where I could potentially have gotten 32. This is allowed because at some moment in time, way back when, we were chased by the lion, and we got to go, and we will take a very small, inefficient amount of energy to keep us moving. Okay? And so you exercise faster than you can get the molecules to oxygen. And this is in response to epinephrine. It can make you go into this state, or if you just work out really, really hard. This lactate is a dead-end product inside the muscle cell, and it will get trapped there. Okay? It will make your muscles tense or cramp up. It'll burn. And what has to happen is your blood has to go through the muscle and extract it, but it doesn't extract it really well. And so a lot of times an athlete will get a massage. And what happens in the massage is that they physically manipulate the muscle, and as they do that, the blood infuses into the muscle and actually allows those really tight bundles to unbind, and what it does is it releases the lactic acid into the bloodstream. If you ever do get a massage, what they tell you to do is drink a lot of water after that, because you flood your system with a lot of these intermediates that are supposed to come out slowly. They're now being released really quickly. The liver can reprocess that lactate to make glucose back, but it's a very energetic process. But we don't lose it, we just have to have it reconverted. We lose potentially a little bit of, well, actually quite a bit of the energy, but we can extract some of it. All right. We really want to get it all the way to the oxygen under aerobic conditions. And we will use the citric acid cycle and oxidative phosphorylation, which are the next 2 chapters, to do that. Pyruvate itself is a molecule that has energy. The first step is a transition between glycolysis and citric acid cycle, and pyruvate is converted into something called acetyl-CoA. We'll actually look at that molecule in detail. Now, again, the problem is we don't eat glucose. What we eat are complex carbohydrates, very long chains of glucose, like breads and starches, or we have disaccharides. And so the disaccharides are broken down, or the long chains are broken down in disaccharides, and we have a lot of entrance of these sugars into either glycolysis itself as glucose, or intermediates that are below that. And so we can see lactose gets converted into galactose and glucose. Now, glucose is the first part, so glucose goes down, glycolysis, that's perfect. Tetralose are 2 sugars of glucose, and it produces 2 glucose, and that goes down, that's no problem. Sucrose produces a glucose that goes down and a fructose. Now, fructose is part of glycolysis, but this is the wrong state. It doesn't have a phosphate on it, but it can be converted. And there's 2 different ways that this one can go. Okay, mannose can be released as one of the in some of disaccharides, and that can be manipulated to enter in as fructose. But what we're going to do is take all of these sugars that are different and put them somewhere in glycolysis to efficiently get them. Once we've got that, we use one pathway to break down carbohydrates. Looking at fructose, there are two ways to do this. All right? One by hexokinase and one by fructokinase. Now, the fructokinase occurs in the liver, and the hexokinase occurs only in adipose tissue. All right? The reason why hexokinase can actually phosphorylate fructose in the adipose tissue is that adipose tissue has almost no glucose in it. And so the hexokinase actually can phosphorylate fructose, but it has a 20 more times affinity for glucose than fructose. So if there's any glucose in a cell, it's only going to bind glucose. Only in adipose tissue, if fructose gets in there, it actually can bind to that enzyme and be phosphorylated directly into a glycotic pathway. Okay, so very rare, but that's how we can get it in by phosphorylation of hexokinase. Most of the fructose is processed in the liver itself. I produce fructose 1-phosphate, which is not one of the glycotic intermediates. And then there is a specialized aldolase that is a mimic to the glycotic aldolase, and it breaks it into glyceraldehyde and dihydroxyacetone phosphate. At this point, that is glycolysis. This is not. But one phosphorylation event, one kinase can get it into the intermediate. So, got a couple other enzymes to get fructose in there. Fructose is important because fructose is found in high concentrations in fruits, and so that's a natural sugar. Okay. Galactose is important because galactose is part of the lactose. So galactose is a sugar, and it takes a little bit to get it in. Now, galactose has a galactose kinase, and it's going to make galactose 1-phosphate. That seems to be straightforward. Okay, this next reaction is weird. Okay, now watch what happens. Galactose 1-phosphate and this molecule called UDP glucose. All right, UDP is another ribose. It's an RNA molecule, just like ATP is an RNA molecule. All right. Sometimes we use the other base pairs as energy molecules, and in this case, it's an activator. What happens is galactose 1-phosphate and glucose UDP react together, and what happens is this hydroxide will react, okay, with this. Let me see. No, it's this phosphate reacts with this phosphate, and what falls off is glucose 1-phosphate. Glucose 1-phosphate is almost a glycotic intermediate. What ends up happening is now, instead of having glucose on the ring, I have galactose on the ring. The glucose can be converted into glucose 6-phosphate. It is now within glycolysis. But I'm left over here with a galactose. Well, I have an enzyme called an epimerase, and an epimer is one hydroxide flipped. And if you take a look, this hydroxide's up, that hydroxide's down. That's the only difference between galactose and glucose. Once I have it embedded in this, it binds into this enzyme and creates glucose from the galactose. And so another galactose comes in, that glucose that has now been flipped is released and can go in. And so I've got this little circle that will move the galactose into glucose 6-phosphate eventually. The UDP portion is the activator, makes a wonderful leaving group. Okay, here's a problem. This is kind of complex, and we see a lot of people that have an issue with enzymes. So lactose has a lot of issues. One, we have a disease, if I can't break down lactose, that's lactose intolerance. But even if you can intake lactose, some people have a problem with breaking down the galactose. Okay? And here's the problem. If by chance I can't do the transfer, all right, then I lose that phosphate and it actually binds into another enzyme, and this only happens when it gets really, really high, and it gets converted into that alcohol. And at that point in time, it's a dead-end product. It can't be processed by any enzyme in the body. And unfortunately, this thing can't be converted, and so it builds high in concentration and it gums up the works. It can get trapped in the liver and become an enlarged liver. It can cause jaundice. Most of the time, after long periods, it causes mental retardation. One indicator also is that this molecule, as it flows through, collects in the cornea, okay? And it can cause a cataract. It's not the only way to cause a cataract, but that is one outcome of this. The only treatment for this, okay, with lactose intolerance, you just eat the enzyme as you eat the stuff. I can solve that problem. The only treatment for this, if we find that a person has this inability or the absence of this enzyme, is to stop them eating galactose. So that means you have no lactose. That means you have no dairy at all. So they have no milk, they have no ice cream, they have no cheese. And that's the only way you can treat this, because any intake will cause a molecule that can't be processed. And so they can't even drink lactated milk because you will have lactose in there. And so we are going to find that in some metabolic pathways or some diseases, that the only treatment we actually have is to limit that particular molecule in the system. But in these, some pathways are like excruciatingly, it has to work or you die. Some of these metabolism diseases, the whole pathway doesn't work, but it doesn't necessarily indicate that it's going to stop production of energy. It just, you got complications with it. And there's just no treatment other than to stop it.